Ladies and gentlemen, please welcome to the stage Dr. Pietro De Camilli.
Good evening, everyone, and welcome to the E.B. Wilson Medal presentation. The E.B. Wilson Award is the highest honor of the American Society of Cell Biology and is presented every year at the annual meeting of the Society for far-reaching contribution to cell biology over the course of a lifetime. Edmund Beecher Wilson was one of the founders of cell biology. He made a number of fundamental discoveries, including the co-discovery in 1905 of the chromosomal basis of sex. It gave me great pleasure to introduce this year E.B. Wilson medalist Mina Bissell, whose work had transformational impact in the field of tissue specificity and cancer biology. Mina has put the field of the cell microenvironment on the map. At the time when the oncogene field was focused on assays that measured the activities of single cell in Petri dishes, Mina looked beyond the single cell, recognizing how the interaction of cells with each other and with matrix proteins can influence cell proliferation, differentiation, and survival. In the early 1980s, she showed that even the activity of potent oncogenes, such as VSARC, is dependent on cell context, and that a single oncogene is not enough to cause cancer. She showed that Rous sarcoma virus will not form tumors in avian embryos, but will do so in chicks. However, when embryo cells are dissociated and put in a dish, they become transformed by VSARC, pointing to the importance of the cell environment. Again, in the 1980s, in a classical paper in Journal Theoretical Biology, Mina proposed the occurrence of dynamic reciprocity between cell and their extracellular environment, with signal flowing in both directions from the environment to the nucleus and vice versa to maintain tissue specificity. In the 1990s, Mina discovered extracellular matrix response elements and the profound effect of extracellular matrix on gene transcription and translation. In a series of beautiful, important follow-up studies, she elucidated underlying signaling mechanism. Over more than 4 decades, Mina's research has consistently been original, important, and innovative. She has raised bold hypotheses, often against conventional thinking, and she has gone on to test them in an imaginative and brilliant manner. Mina is a distinguished scientist at the Lawrence Berkeley National Laboratory, where she spent her career. In 1988, she became director of its Cell and Molecular Biology Division, and in 1992, director of the entire life sciences of the laboratory, a position that she held until 2002. In 2012, the Lawrence Berkeley National Laboratory honored her with their inaugural Lifetime Achievement Award. Mina was born in Tehran, Iran, and after high school, she came to the United States to study at Bryan Moore, coincidentally the same institution where E.B. Wilson served as professor of biology in the late 1800s. She carried out the rest of her studies at Harvard with degrees in chemistry and bacterial genetics. You can read about her more interesting and intensely lived life in the engaging essay that she wrote for last month's issue on the molecular biology of the cell on the occasion of her receiving the Wilson Award. And in that issue, in that essay, you will find also a beautiful picture of her extended family. Mina served on a wide variety of national and international advisory board, and she's an influential, passionate, and effective advocate for young scientists. She's been heavily engaged in ASCB activities, and in 1997, she served as ASCB president. She has received an impressive number of honors, including one named after her. She's a fellow of the American Academy of Arts and Sciences, the American Philosophical Society, the National Academy of Medicine, the National Academy of Sciences, and the Royal Society of Chemistry. As many of you who had the privilege of working with her or who know her, as many of you had the privilege of working with her or who know her, she's the most engaging and vibrant person and a wonderful mentor. Her enthusiasm and passion for science is an inspiration for young scientists. Please join me in congratulating Mina for receiving this award.
Thank you. Oh, we have to come here. Thank you. Thank you. Well, thank you very much, Pietro, for this lovely introduction, and thank you, Peter, and thank you, ladies and gentlemen. I'm quite honored and humbled with this award. Those of you who have not heard me have to stay awake, and the rest can go to sleep for the next 7 minutes. I have to give you the background so that you could understand what I have done with my life, and I have very little time. This is a picture of human breast. Look how gorgeous it is. It has 12 different cell types, and I wish I could show you all. 12, not 4, 12. And it's all put together in this picture that Kurt Heinz took. And the question is, how does this breast remember to do all this, and then to do all the rest of those functions? So I like to start out with this huge question. How do 10 to 70 trillion cells, did you know you have so many cells? They all have the same genetic information. Now, did you know how many zeros 70 trillion cells has? You can count them fast. All right. Now, I used to say that there is no such thing as terminal differentiation, and that you have to think about how your nose is your nose and your elbow, and people would laugh. Now, one morning you got up and you saw this stuff about Dolly, right? It was a single cell from the mammary gland, and it had all the information to make a whole sheep. So I rest my case. How is tissue specificity, noseness, breastness, prostateness maintained? How does one study the problem in mammals, especially human? I wish Drosophila had a mammary gland. I can't talk about the rest of that stuff. So we chose the mammary gland as an experimental animal, and thank you, Pietro, for that very nice introduction. You told them some things about before that I don't have to show. And it develops again and again and again. And here is a human breast with one of those lobules. We have about 4 or 5 or 6, there's a debate. And we wanted to say, how does the mammary gland know to be a mammary gland? And we didn't know what to study. The nipple is all the way back there. I don't know if I have a pointer. If I don't have a pointer, it doesn't matter because, okay, let's see. I can't do both. It's too many things. Okay, so we then decided that this has, the nipple is in the beginning where the baby sucks, and then the primary branch, secondary branch, tertiary branch, they do all sorts of things. So what should we settle on? And we settled on this thing called an acinus. Now, you heard that I was a chemist and a bacterial geneticist, and I knew no biology, and I'm not proud of it. But there was one good thing about it, and that was the fact that I wasn't corrupted by dogmatic statements in the field or dogmatic textbooks. So we said, and I knew, honestly, so little. Here is the, oops, let's see if I can get it back. Here is the one layer of beautiful epithelial cells. They are polar, they are secretory, and then around on the blue, there is myoepithelial cells that squeezes the gland and where the baby sucks, and then there is this other thing around it. So we said, even in vivo is too complicated. We should put these cells in culture and ask them, how do you do it? We put it in culture, and it was a disaster. Here is a single mammary epithelial cell, beautiful polar. It has secretory activity. It has all this gobs and gobs of milk that is happening, gorgeous nuclei, fat droplets, secretory, polar, basal, apical, everything. You put it in culture, and literally, in about a day or two, it looks like what's on the right. Polarity is lost. Look at this, look at the signature look at the way the nuclei look. Look at the one on the right and compare it to the left. Everything changes, and they forget to make milk. And you say, I gave you all the lactogenic hormone. What's your problem? Well, they can't make milk. So we section the mammary gland of the mouse. Around each mammary gland, we said there has to be something that is not in culture, right? So the physicists would say that, the high school kids would say that, but I couldn't get that out of a lot of other people, that this thing, which is called extracellular matrix, these huge molecules, in fact, is out there, and we destroy it when we put the cells in culture. So I said, my goodness, it has to signal. They said it can't, it's insoluble, it's huge, it's outside. Well, I said it has to signal. And what we did was that we said we want to model this, and to make a very long story short, a number of people had defined this thing called Matrigel, of which a lot of people have made a lot of money. It's a gelatinous material. It's like a gobs of basement membrane. You put the cells in them. In a paper in 1989 in differentiation with Mary Helen Barcelotov, who was my postdoc and now is professor at UCSF, she put the cells on here, they pull it over them. In 4 days, they look like that. Next to it in the middle 3D culture, even the sizes are the same to the one on the left. The sizes are the same. I mean, imagine. So we said, do you look so gorgeous, or do you actually make milk? So we made some antibody against milk protein, and there it is. We got milk, and I had the red is milk, the blue are these beautiful structures, and gobs of milk in comparison to the 2 counts of radioactivity we would get in 2D. So I had one of these big birthdays. They put a milk mustache on me, and they took a picture. So I'm not showing it to you. Okay, what about humans? What about humans? Ole Peterson came to my lab. We started working together, and I said, you're an MD PhD, Ole. He was an assistant professor in Denmark, and he said, I like what you do. Can I work with you? I said, well, no, you go back to Denmark and get some reduction mammoplasty, where women do it to themselves, and we can get this thing out of the garbage and bring it and separate the cells and try and find out whether or not they did what the mouse did. So here it is. On the left is that gob of fat with the stuff inside. These are when we dissociate the cell, looking like everything that you know about. On the right is when we put them in this matrigel or laminin-rich gel. For once and all, I have to tell you, you don't need matrigel. What you need is laminin and a gel. So it could be laminin in collagen gel. All right, come talk to me afterwards if you want. So don't tell me matrigel has all this junk in it. It doesn't matter. It overrides because it has the right property of the gel, and it has laminin. Okay, so I said, if the normal cells know how to do that, on the top, those are not single cells. Those are 3-dimensional structure that you see on the left. Those beautiful cells with propydiumidite and green actin. And these are tumor cells. And all of a sudden, we have this gorgeous assay for human breast tumors distinguished from normal. And we can do this in 7 days. We now, these days, can do it in 4 days. We used to have to inject them in the mouse, wait months and months, and it's absolutely beautiful, and they are clonal. So if you take a clone out, you can do all sorts of things. You can propagate it, you can do cDNA libraries, you can do whatever people are doing with their wonderful molecular biology. But folks, really, this is the Society of Cell Biology. And of course, I'm prejudiced. But cell biology is where the biology is at. Everything else to me, biochemistry, molecular biology, et cetera, these are technologies that we could use to understand cell biology. It's an exciting field. It never ceases to excite me. Okay, so then I wanted to compare why the normal knows and why the tumor doesn't. And so what we did was that we took cells from reduction. I didn't. Ole did, and his professor did, from reduction mammoplasty of a woman. They put it in culture with no antibiotic, no serum, only EGF, grew, and then they removed EGF, grew, and then they injected it into the mouse, and eventually they got a tumor, which is here. And if you look at those cells, which were pre-malignant, and then the malignant on the bottom and the normal on the top, and look at them under microscope, they all look the same. But if you put them in 3D, which is now, I drew it for you, passage 60, P53 is gone. There is deletion, addition, hundreds of mutations. Look at it. The cells think they're normal. Then MYC amplifies. They still think they're normal. Passage 175, God knows. Passage 500, God, they still think they're normal. They form these gorgeous structures. And when you look at the cells where EGFR was removed and was injected into animal, they have a lot of mutation, but they still are not malignant. But the beauty of it is the structure goes before malignancy sets in, okay? When the cells become malignant here, they are different from that. That happened once in a blue moon. But we have a model of breast cancer outside humans is starting from a reduction mammoplasty. It's a very exciting model. So I started saying the chicken that you heard about and the embryo, when you dissociate them and put them in a dish, they lose it. They express things that they wouldn't express in the embryo. So I said, if form and function are related, and if everything depends on its architecture, we should be able to take a tumor cell that makes those ugly tumor-like stuff, find a way of organizing it so that it looks normal, then the cells should think they are normal. They shouldn't form tumor. Alternatively, if you take a very, very normal mouse and do make a transgenic mouse that you can go in the middle of pregnancy and destroy the architecture, without giving them oncogene, without taking away the suppressor gene, then they should think they are malignant. And that we did with Zina, but I have no time to talk about it, but we did, I'm showing you the human. Non-malignant, malignant, came from the same individual. So Valerie Weaver, who was a brilliant postdoc and who has her own little empire at UCSF, came, and we had done little studies before where we could revert the cells, but they weren't so well done. And she found out that, in fact, when you look at the normal surface of those cells as opposed to those, they have all the receptors, but their balance is gone. The tumor cells are high, the normal cells are low. If the normal is 1x, tumor is 5x, 6x, 7x, and we used inhibitory antibody against integrin to actually ask the question, could we bring it to 1x and see what happens? And this is what happens, and as you see, it was a long time ago. It came out on the cover of Journal of Cell Biology. We were very proud of it. We didn't send it anywhere else, and we were very glad they put it on the cover. And to show you something else for you to think about is that those cells have the same genome as the malignant cells, but they look very different. This paper came out last April in Science, and it's the opposite of what we found. I don't have time to show you the rest of it, but what it is, they took cells from the skin of normal people, and they found out they had all the oncogenic pathway. So as you're sitting there, and you're my age, you are full of oncogenic mutation, and yet, thank goodness, we still don't have cancer, or we don't have malignant cancer. Why? I wrote an article in Nature Medicine called, Why Don't We Get More Cancer? And you can look at it. Okay, look how beautiful they are. Non-malignant, beautiful polarity, malignant, reverted. They are not 100% normal, they have mutation. but it's lovely. Okay, we use this, and as I said, every one of them does it. Those are malignant on top. When we add them beta-1 integrin, etc., they get reorganized, and people still wouldn't believe it, so we had to show that reversion was reversible. So what we did, what Valerie did, was that she took the cells without antibody in the gel, with antibody in the gel. We took these cells that are, I should actually do that so people on the other side of the audience don't feel neglected. So here is, on top is malignant, on the bottom is treated with beta-1 inhibitory antibody. You take the cells on the bottom, dissociate them, put them in a dish, and the one on the bottom do that, and if they give them the antibody, they do this. And what does this say? Phenotype is dominant over genotype. Now, it used to shock people, and I would say, go look at yourself in the mirror. The same gene, the same gene, the same gene. Why are you so surprised? This makes a lot of sense. You can have this. Okay, so if you are actually having form and function related with together, you want to actually ask the question, how do these signaling pathways integrate? So when we do beta-1 integrin, in 3D, EGFR goes down, MAP kinase goes down, PI3 kinase goes down. If we put this through glycan, et cetera, they all talk to each other to integrate, while in the tumor, they don't integrate, and in 2D, they do not integrate. All right? So this model is like the eye of the Drosophila. It's so robust. And if you go from non-malignant to malignant, you have hundreds of steps. But when you go from a single malignant to a three-dimensional structure, you're reverting it in one step. So it can be used to understand how architecture is maintained. Okay, this thing rocks. Without this, you would not be here. Without this, you wouldn't be living. It's as important as P53, and I'll show you why, and if I have time. So it is really the most important component that actually does a lot of things from outside the cell talking to the nucleus. Okay, now the ECM is outside, the nucleus is way up there. How do they manage, and how does this architecture get formed from a single cell? So here is the beauty of one of these things. You see how gorgeous it is? How is it formed? So this lovely girl whose picture I just discovered 2 minutes before I walked into the podium is Candace Tanner. I hope she's in the audience. She now is a professor at NIH. So she is a physicist. She came to me, and believe me, there are a lot of physicists who don't know anything, and they think they know a lot. And I have had a lot of them. But this one was really smart. So she said, when you put the cell in there, what happens? I said, I don't know. She said, I'll find out. Now look at this. She puts either a primary breast cell or a cell line that is of the breast in this gel, and look what happens. Did you know? This explains why women can have babies again and again after involution. It's just like an embryo. It reorganizes, and it makes this gorgeous three-dimensional structure. That was in 10 days. Okay, now you do this with tumor cells, and look what happens. The normal cells are attached together. The tumor cells are an absolute mess. They don't talk to each other. When that cell becomes one, it then becomes polar. Then it attaches together, and that scaffold serves for the rest of the architecture. And I predict that what Candice discovered in the breast is going to have different kind of movement for your mouth, for your eyes, for your brain, for whatever. But it's remarkable. They are attached, and one cell in here becomes two, and two becomes three, just like a stem cell. You put them in a gel, and they think they have to make another breast. So that's what they do. Okay, so you remove adhesion molecule, no, you remove the polarity molecule part 3. In the normal, you see how beautiful the movement is. In the malignant, not in the malignant, in the one that you have taken the PAR away, you see the movement. So we do this with E-cadherin. On the left is normal, gorgeous. The movement is lovely and tight. Coherent angular motion, PNAS 2012 or something. And that one in, we have removed E-cadherin, and look what the message is. Okay, now, we now know that you put a single cell, it starts moving, and then makes this gorgeous structure. The question is, why do cells stop? Why do you give them laminin, and they actually think they should stop? How is the mechanism? So we find actin in the nucleus. It is absolutely real. A lot of people are beginning to find it after years of saying this is an artifact. The work of 2 different people. The first one is Spencer. She wanted a green card, and she wanted a fast publication. And we sent it to Journal of Cell Science, a wonderful journal, and the editor was so excited that she immediately published. So depletion of nuclear actin is a key mediator of quiescence in epithelial cells, and it all depends on laminin being present. So you do that, you see that the nuclear actin is up there, and it starts getting out. She did it in 2 hours, but Alex, oh, his name is so hard. I don't know if I have it there. I don't. Terrible. He's in Brazil. He's now assistant professor. He could show it that is in about 30 minutes, actin exits. And you see that over there. The loading control is good. I have so little time to show you beautiful biochemistry. But anyway, you see it. Now, here are the 3-dimensional normal. There is actin there in the nucleus. You need some actin for transcription. Actin complexes with Pol2, Pol3, etc., all sorts of transcription factor. I love to do something with Ivo Nogales to find out what those transcription bundles do. And look at this in the malignant. Look at the level of nuclear actin in the malignant. And we did all sorts of studies, bleaching the cells, finding the rate of the exit of actin. The rate of entry doesn't change, the rate of exit. And we have a lot more data, and hopefully will come out in cell report. Now, we add the cell, it does this. The signal goes to actin, the cells stop, become polar. But when we reverted, remember I showed you that scheme, that the beta-1 integrin was down-modulating EGFR. EGFR was down-modulating this, and that, and that, and that. And we did arrays for all of them. And this gal said, they all revert, but the pattern of reversion of the arrays that we did looking at gene expression is not the same. So another postdoc did an analysis and showed that there are about 70 to 150 genes that they have in common. Okay, so what she did, she said you have to have microRNA, and they are linked to phenotypic reversion of the human breast cancer cells. So you remember that model I talked about, so we are again going this way. So here are the different kind of revertants with different inhibitors, etc. And here are all those different revertants. Now she took and she said, these things that are in common, which microRNA actually could regulate them? So she did a microRNA library, whatever it is you do these days, and she, as you see, I can't describe in detail. This is not published. It just went to eLife. So I'm going to show you a lot of unpublished data, and the last part of it is so exciting. Don't go and do it, please. She'll kill me. She's now an assistant professor, and she needs her publication. Okay, so she found 10 microRNA. She found 3 of them that really work well in reversion when she put them in. And then she took the human breast section, and what she showed was that these 3 microRNA were lost in malignant cells. Are you still with me? I'm trying to go so fast. Yeah, it's okay? All right. At least one person says yes. Okay. So, what do these things do? One of them, put back, brings up the other 2, and brings up everything else. So she went up and she went down, although in my book, up and down don't have meaning. It's all dynamic, it's reciprocal, it's a closed circle that gives you the shape and the form and function of your tissues. Okay, what is up there? In order for them to revert, they had to inhibit these 2 nasty transcription factors that each one of those will inhibit those transcription factors, and these transcription factors upregulate MMP9. And what does MMP9 do? Destroys laminin. We have a paper in Gene and Development. We show that when we inhibit MMP9, we can revert them, and there's a whole lot of pathways, etc. So you want to have those inhibited. Once you add laminin, that happens. Okay, what's up there? HOXD10 needs to go up, NF-kappa B needs to go down. If you put NF-kappa B, you're not going to get the closure of the differentiated state. If you don't upregulate HOXD10, you're not going to get it. Now, how many papers have you seen on P53? 90,000, 900,000, God knows. Has anybody actually taught what in vivo, in the different tissues, activates P53? Well, P53 regulates those 2, and above P53, what on earth goes on? What are the things that go from laminin 1 to laminin 5? P53 gets regulated by laminin 5, and P53 regulates laminin 5 because it binds to its promoter. It's all in that paper. It was so big that people kept saying, cut it in 2. That's why we went to eLife. Bless their heart. They are willing to do, we hope, if the reviewers do it, a complete story. It took 6 years, or now it's 7. Okay. This is what I don't want you to go and imitate. Between ECM outside and P53, there is nitric oxide. You add laminin, nitric oxide goes up. Without nitric oxide, your tissues don't do anything. So that's what we have. NO is there. And other factors come after NO. But let me at least give you a little bit about NO. Okay, the NO regulates ATM and P14ORF and P53, etc. And this cycle gets closed this way. But let's look at what NO is doing. It's absolutely mind-boggling. Well, this is something that we put together from many different kind of papers. We did glucose inhibition, we did this, we did that. We don't have time to go through it, but they are all connected, and they all are operating in 3D, and there are some new pathways that are not in 2D and vice versa. Okay. So we asked the question, does NO really matter in vivo? So these are primary human cells, a chunk of them from reduction mammoplasty. Look at that. It has the vehicle. It branches magnificently. We inhibit it with an inhibitor of NO, and look at it. Doesn't that boggle your mind? It absolutely boggles mine. Okay, so here is the most interesting part of this. The tumor cells on the bottom don't induce NO when you add laminin. It's the same thing with actin. The tumor cells do not respond to laminin by pushing the actin out of nucleus. So the pathway between NO and actin is what's gone bad in tumorigenicity. And these are the issues that we don't even see when we work in 2D. So here you are, malignant cells don't make NO. We give them NO, they revert. MCF10A that are a cell line, they do it. The cell lines that we got from humans really rock. They have beautiful NO. There's a whole lot of other things to tell you, but what I want to end up with is here is a picture of Manfred Auer. He and I have been collaborating for 12 years. He has been working with Zeiss. He's been working with all these lovely people who are actually, some of them are sitting on the front row here. Some of my postdocs, wonderful people. None of this would be possible without them. So I said to Manfred, we need high resolution. And I want to see whether or not there is in fact interaction between the ECM and the cytoskeleton and nucleus. And here is what we have found. This is actually the work of Claire Robertson, who unfortunately I had to take a lot of the other stuff out. We noticed that under these conditions, you see that little thing in the nucleus that looks like a little finger going in. And Manfred took this to Zeiss. They did a lot of bioinformatics, a lot of analysis, and it's going to be in the picture of Journal of Cell Science, which is actually on the web already. Those of you who are interested in structural biology, please look at it. And it's a beautiful volume, and that various people in the audience actually have worked on it. And this gal, and Jamie Inman is my lab, and Danielle was graduate student with Manfred. They have contributed in many different ways to this. And here, excuse me, can you push that button? Thank you. Look at this. That's a single nucleus, okay? It's not two cells. It's a single nucleus. Look at that. That's the cytoskeleton that goes from one end of the nucleus to the other. It doesn't pierce the nucleus. It's like a donut, okay? It goes like this. Sometimes closed, sometimes a tiny bit open. Now that even boggles my mind more. Amazing. And all of this happens when we add laminin from outside. This was in 3 dimensions. These are the different undergraduate in my lab. We had her picture and somehow came out. Cerise. Cerise, are you here? Anyway, she did this beautiful rendition of it. So we went to this guy, Koo, in Berkeley, who does storm, and we asked the question, can you look to see with these antibodies in high resolution whether or not what we are seeing is in fact cytoskeleton? So he shows in that very beautiful, the existence of SUN1, which is actually can talk to various aspects of chromatin and pankeratin and the rest of it. And here is another one in higher resolution. It's really, really exciting. Now imagine how much there is to still discover and how little we know. We know a lot, but we know very little. And one of the biggest things I say to people is, don't get arrogant. Allow yourself the passion of saying, I don't know, what does this mean? So I say to the young people, trust yourselves. Look at your own data, make your own decisions. One life, do something good with it. So here is my dynamic reciprocity. Jamie Inman actually put this thing together. Let's see, there is integrins, there is laminin down here, and here are the signals that go back and forth. And next to it, you have this beautiful bundle and a tunnel in the middle of the nucleus that the cytoskeleton finds and goes through. Imagine how much we have discovered about how that process works. Here are the cytoskeleton that comes from out there, signaling from ECM, going all the way to where sun proteins are. and coming out at the other end. Okay, so I'm almost done. And many of you have seen this. Here is water coming into the shore from a NASA satellite. Here is coral. And I didn't show you, we have beautiful papers on metastases, on dormancy, on God knows whatever. But here is a picture of the mammary gland of the mouse. Now, do they look similar? So I say that we know everything about human genome. We know the language of the genome, the alphabet of the genome. We know everything, the grammar of the genome. And every time I ask people, how do you describe the fact that the genome here and genome here, they may be edited, but they are single cells that are edited, not the tissue. How do you explain? Just by sequencing more and more. It's perfectly good, but it doesn't answer a lot of questions. So I would say to you that we know very little about the language and alphabet of form. I mean, imagine how much we have to discover about your mouth and liver and kidney and this and that and brain, God knows. So a whole new horizon for the young and the passionate old, and that's me. Go to it. Don't say these famous people did it. The famous people don't know what they're doing. And I love this cartoon. Again, many of you have seen it. I keep showing it. So here is the cat, and there's the authorities. And the authorities is telling the cat, don't you dare think outside the box. Now, I used to be the cat, very rebellious. And those were the authorities. And here I am giving the Wilson Medal lecture. It means I became an authority. So don't believe me. Do your own thing, okay? And here is Candace. Here is Saori. Here is Ginny, who did the acting. This was a few years ago. And then here are my group in 2012. Then I lost a lot of money. But most of them who have left are either professor or doing what they like. And they all deserve a circle. They're all amazing. I owe them so much. They cheer me up. They keep me young. And it's a pleasure to work with them. And you see, I'm the only one who doesn't have a halo. Okay. And here is the beautiful dome at Lawrence Berkeley Lab, where Eva Nogales did a lot of her pictures that she showed you yesterday. She was a postdoc at Lawrence Berkeley Lab, so here we are, 2 women from Lawrence Berkeley Lab. Yay. We are here on Porter Lecture and Wilson Award. You do the path of, you do X-ray analysis and soft X-ray and this and that. And they took the dome of the old place where the atom got dispatched, and it was done as the first lab in US was Lawrence Berkeley Lab, and so many people mixed us up with Livermore. And I hope you forgive me, an old lady up here who had to tell the physicist how to not mix us up. I show this when I go to these other national labs, and I said, even Harold Warmus mixed us up, and he's at UCSF. So those of you who don't know, Lawrence Berkeley Lab is a campus of University of California on the top of Berkeley Hill. It's separate and is interacting with Berkeley. That's one of the reasons I stayed there. We have engineers, we have physicists, we have chemists. And when I wrote my paper of dynamic reciprocity, I said, we need them all. Life is complicated, and that's the beauty of life, that is complicated. But we need them all. We need all these capabilities. And look at this beautiful thing. I say, Livermore doesn't have that bridge, that view, and what does this dome remind you of? A breast? Okay, only women get away with this, so. And the physicists say to me, Mina, darn it, we used to come here and look at this. Now we can't think of anything else except rest. I say you could do worse. It's okay. Thank you very much. Now, I don't answer questions? I guess not. All right. Yeah, I'm free to go. Yes? Okay. Very good. And can I take my medal with me? Yes. Thank you.